All right. Thanks for coming to our webinar. My name is Ramji Srinivasan, and I'm co-founder and CEO of Teiko. I'm going to present to you our team's data on our CLIA validation for our pan-immune profiling test. So we'd like to say we put the precision in precision immune monitoring. And again, you can read along at home. We've written a method validation plan and a method validation report that's available online about our pan-immune profiling test. So the problem we're going to talk about today is about high stakes reliable measurement. Now, of course, we're talking about the immune state, but to ground your intuition, let's talk about an easier to understand problem. Let's take weighing a crying baby at low birth weight. Imagine you've got a baby who's just been born, and the problem statement is that low birth weight babies that don't gain weight quickly need medical intervention. So you've got a baby born at baseline, she weighs 2.115 kg. So the first thing you need is food to make sure she gains weight. The second thing is that you need is a scale to make sure that she is in fact posting that weight gain. So ultimately, just to measure one variable, you need a weight scale that can reliably detect this weight gain, because if she's not gaining weight, she might need medical intervention. So if you were to take multiple measurements from that same baby, measurement 1, measurement 2, measurement 3, these should all be around the same place. Remember, her true weight after 1 day was 2.165 kg, and the baseline weight is 2.115 kg. So now think about the immune state. We just talked about 1 kilogram, 1 measurement. If you are to translate this to measuring the immune state, imagine you've got a patient and she's drawing blood, and you get a baseline test for her and look at her immune state. You're looking at T cells, B cells, NK cells, monocytes, dendritic cells. Now, if you give her a drug, and on day 1, all those immune cells will shift. The T cells, B cells, et cetera, these will all move, and they may be in different activation states. They may be in different amounts. So similarly, you need an immune scale that can reliably measure immune response across these different cells. So for measurement 1, measurement 2, measurement 3, as a simplification, you would have to look at these dozens of different immune cells across hundreds of thousands of cells for every sample that you take. And each of these matrices should look more or less similar to one another. So what we tested at Teiko is peripheral blood mononuclear cells isolated in-house from healthy donors. So we got healthy blood donors, 9 of them. The blood was sent to Teiko. We did isolation at our lab, and then we used these PBMCs in our tests. And our immune profiling test covers many different immune compartments simultaneously. So not just T cells, but B cells, NK cells, monocytes, dendritic cells, and many different subtypes. For example, classical dendritic cells, CD16 minus NK cells, et cetera, et cetera. And if you zoom into that blood sample we talked about, underneath the hood, you can be looking at dozens of different markers, in our case, specifically 40 immune cell markers from CD3 to TBAT, and then hundreds of thousands of cells for every sample. And then in this big matrix, you have, say, a signal intensity. So for example, cell 1, CD3, cell N, CD3, you would have a signal intensity. So now you've got about 8 million data points to look at, and each one of these 8 million data points should look similar from run to run, from person to person. So again, going back to our question, is measuring the immune state from measurement 1, measurement 2, measurement 3. If you run these through what's called a mass cytometer, you get a big matrix, about 8 million data points, 40 times 200,000. And those 8 million data points should look similar from run to run. So the big question our customers in biopharma have is this thing valid? You're measuring so many things simultaneously. Can you confirm that this thing actually is reproducible? Because we're going to be running clinical trial on it. And so to answer that question, we wrote out a big validation plan with 4 key measures: intra-run, inter-run, interoperator, and stability. Intra-run precision means take the same sample, split it up across the same run. Inter-run means the same sample across different runs. Interoperator is taking samples from 2 donors and fixing them by different operators. And stability is taking the same sample and then comparing it across time. And each of these have an acceptance criteria measured quantitatively by what's called a coefficient of variation, which is a measure of variation between, say, replicates or runs. And for stability, we measured the change between days. So we're going to go through these numbers on each slide. We looked at the immune subsets analyzed, the average coefficient of variation, and the average change. And just as a note, we only include populations with over 100 median cells to smooth out variability. So just a quick tutorial on and a reminder of coefficient of variation. Going back to the baby example, her weight is 2.165 kg after day 1. Her baseline weight is 2.115 kg. So imagine you have a good scale that is measuring her weight reliably. Just by eye, you can see that each measurement over here, 2.16, 2.22, these all seem around the same range. And the coefficient of variation, therefore, is quite small, 1.44%. It's computed just by taking the standard deviation across these measures and dividing by the mean. And this is a bad scale that swings all over the place, 3.66 kg, in fact, posting a lower weight gain. This is a coefficient of variation of 46%. So this is not a good one for measuring a baby weight. So now remember, we had those 4 measures: intra-run, inter-run, inter-operator, and stability. Intra-run is taking the same sample, splitting across the same run. And for this, we posted not just by major lineage, but in the detailed report, you can see by the different subtypes. And our average CV was 3.52% versus our 25% criteria. So really strong performance across all the major cell types. The second one is inter-run, and we took the same sample across different days. We took a blood sample on day 1, then across day 2, day 3, and then we compared these analyses. And again, the major lineages look great. So the average CV was 6.66%, and we set out a 30% CV acceptance criteria. So dendritic cells, monocytes, et cetera, these were all very low CV. So it was good to see this. And then we looked in more detail. We did this in the more detailed report, but I'll just show this for inter-run precision. We looked at the cell population size. And so, for example, CD4 plus TEMRA, the x-axis over here is cell population size. The y-axis is coefficient of variation. So these are smaller cell populations, which you'd expect would have more variability. And as we saw, larger and larger populations have less and less variability, which makes intuitive sense. A cell population with fewer cells is going to be more variable because you're going to have fewer of them to pick up. And then the third comparison is inter-operator. So we did 2 donors and we fixed them by different operators, and then it was ultimately stained by a single operator. And we compared across these by major lineage, and again, found single-digit coefficient variations for both these donors. So 4.83 for donor 1 and 7.36 for donor 2. And that we compared to our 25% CV acceptance criteria. And then the last item is stability. So we took the same sample, we processed after 1 or 21 days. So took a sample, put it in the freezer, then processed it on day 1, or took the same sample and then processed it on day 21. Because a lot of customers will send us a sample, but they don't want to process it right away. So for this, we looked at the stability by major lineage, and we saw, again, the average percent change was very small here, which is good, negative 0.01%. You can see the detail in here by major lineage: T cells, B cells, NK cells, monocytes, dendritic cells. All these are quite stable across the board. So again, the big question that customers have is this thing valid? And we looked at all these 4 measures: intra-run, inter-run, inter-operator, precision, and stability. We set out different criteria, and we concluded yes. For intra-run precision, if you remember, we set out CV of less than 25%. We posted 3.52%. For inter-run, we set out less than 30%, 6.66%. Inter-operator, it was less than 25% and was 4.83 and 7.36%. And stability less than 25%, or excuse me, plus or minus 25% between days, and it was 0.01%. We come up with these CV ranges after looking at the flow cytometry literature extensively, which was in this 25 to 30% range. So that's how we came up with these acceptance criteria to begin with. So ultimately, the conclusion is we believe that we're well suited for demanding clinical trials. And if you're interested in the validation plan or validation report, feel free to download at home or ask any questions in the webinar. So with that, I'm going to pause and see if there are any questions from the audience. Okay, so first question is, what kind of data do you have for whole blood? So we don't have data on whole blood yet for this particular assay. We've done this on peripheral blood mononuclear cells, but that's ultimately in our game plan. And we have also introduced a Toku kit, which makes whole blood collection easy. That's available on Teiko.bio. But yes, that's a great question. This data set is only on PBMCs for today, but eventually we plan to do this similar analysis on whole blood. Okay, next question is, were the PBMCs created in-house or from a lab? And the answer is a mix of both. The healthy donors were sent to us from an external facility, and then we turned them into PBMCs at our lab. So just want to poll to see if there are any other questions, comments, statements. All right. Well, if not, that's going to conclude the webinar. And really exciting to have met everybody. And if there are any questions that come up afterwards, you feel free to contact us at my email is ramji@teiko.bio or info@teiko.bio on the web. So thanks, and we're going to sign off soon.